Today I will talk about biochemical mechanisms associated with nucleotide analog viral polymerase inhibitors, and with specific focus here on remdesivir and its mechanism of action against SARS-CoV-2 RDRP. So, disclaimer, I received funding from Gilead Sciences on biochemical studies on the mechanism of action of remdesivir. I studied chemistry back in Germany and did my PhD at the Max Planck Institute for Biochemistry. And this is where we started to work on HIV-1 reverse transcriptase. Later, I moved to Montreal, did my postdoc in Mark Weinberg's lab, and got interested in drugs, small molecule interactions with HIV, reverse transcriptase, and also drug resistance. And I started up my lab at McGill, and we really focused on HIV and later on hepatitis C. All polymerases and nucleotide analogues and a couple of other classes of polymerase inhibitors. Then in 2014, we moved to Alberta, to the University of Alberta in Edmonton, and we changed directions. We felt that we can't really contribute anymore to drug discovery, development, and mechanism of action and resistance to drugs associated with HIV and hepatitis C. So for Hep C, we had sofosbuvir and a number of other drugs that were evaluated in clinical trials and approved at around that time. And they were highly potent, and we just felt we need to move in an area where we have medical needs and where we do not have adequate medical countermeasures. And these were emerging pathogens and like Ebola, coronaviruses, and so forth. Now, if I go back to HIV reverse transcriptase, this is a structure of the enzyme, and it is folded like a human right hand with thumb, fingers, and palm. The active site is the palm, located in the palm region, and the nucleic acid is held in place by fingers and thumb of these domains that are indicated here. Here's the nucleic acid. Most polymerases have this general shape, like a human right hand. Sometimes it's a little bit more open, sometimes it's closed, but in principle, the subdomains of polymerases are divided as such. This is the basic function of the polymerase active site, and I just go briefly over this. There are some important features to understand that are required to understand the basic functions. And there are two divalent metal ions at the active site, two magnesium ions as indicated here. And they help to position the 3' end of the primer that's indicated here by the blue or cyan nucleotide. And the incoming nucleotide is here shown in green with its triphosphate moiety. So you can see that there's interactions with the two divalent metal ions, and what happens at the active site is there's a nucleophilic attack of the 3' hydroxyl to the alpha phosphate of the incoming nucleotide, and pyrophosphate is released, and the primer is extended by a single nucleoside monophosphate. So that's the general mechanism. It is important to show that because nucleotide analog inhibitors like remdesivir, and I will talk mostly about remdesivir, but many others, all others, they bind and compete with the natural nucleoside triphosphate here at the active site. So the competition with the natural counterpart is extremely important to understand the mechanism of action of these compounds. This is the first one, the MOA, so mechanism of action of AZT, the first nucleotide analog or nucleoside analog that was approved for the treatment of HIV in '87. And a little bit nomenclature, so you call it nucleoside if there's no phosphate attached to the 5' end, and then the nucleoside analog is then intracellularly phosphorylated to the triphosphate form, and the triphosphate form is accepted by, in this case, HIV-1 reverse transcriptase. The monophosphate is incorporated and pyrophosphate is released. So that was, in the old days, the mechanism of action of nucleotide analog inhibitors. It was chain termination. So they get mixed up with the natural counterpart. In this case, it is TTP, get incorporated and cause chain termination. And why? Because the 3' hydroxyl that is required for the nucleophilic attack on the alpha phosphate of the next nucleotide is not present in these chain terminators. So there's a chemical reason for that, why we observe chain termination. So here in this case, for AZT, it is a 3' azido group, and there for 3TC, you have a sulfur and a bunch of other variations. The bottom line is always you lack the 3' hydroxyl group, and that usually translates in chain termination. Now, moving back now or forward to 2014, there was the West Africa Ebola outbreak, and that really motivated us to work on these emerging pathogens like Ebola, where we do not have any medical countermeasures, no polymerase inhibitors, and that obviously is a logical target. At that point in time, believe it or not, the polymerase was not even available. It was not published. There was no protocol available of the Ebola polymerase. So it was difficult to identify compounds that would inhibit the virus via inhibition of the polymerase. At around the same time, then the WHO came up with this blueprint, and they listed priority diseases for which no medical countermeasures were available, or no adequate countermeasures. And the list is quite long, with Crimean Congo, Ebola, Lassa, the coronaviruses, Nipah, Zika, and so forth. And you have here disease X. This is the unknown virus that may cause outbreaks or pandemics, and obviously that was SARS-CoV-2 and COVID-19 in 2019. So going back then to 2016, roughly, in the aftermath of the Ebola outbreak in West Africa, Gilead teamed up with CDC, USAMRIID, and they screened the Gilead library for nucleoside analog inhibitors against Ebola, and identified a hit. And this is a nucleoside analog with a 1' cyano modification that was reported earlier at around 2010. As being active against hepatitis C and some other viruses, including SARS as well. Gilead prodrugged the compound, and this is Remdesivir, and previously GS5734. That was, published also in Nature 2016. Very comprehensive study on the antiviral effect in cell culture in animal models against Ebola. Here is the intracellular metabolic activation in greater detail. Again, you see here the prodrug. It is hydrolyzed to the monophosphate version, and then eventually the triphosphate is generated, which is believed at that point also to be the substrate for the viral polymerase. The question for us was then, how does it work? And there are just some features here that are nicely shown in a cartoon from Kathy Szalay-Radke, and you can see the differences to the natural counterpart, adenosine. This is first of all the 1' cyano group. Then I have to point out here, this is important in relation to what I was mentioning regarding AZT. Here, we actually do have a 3' hydroxyl group. So in other words, the mechanism of action is not necessarily chain termination, because if this nucleotide goes in, it can attack the next incoming nucleotide, and RNA synthesis could theoretically go on. This is the prodrug moiety, and there are also some modifications here in the base moiety. So we looked at Ebola, at the Ebola polymerase, and we expressed it. It's a complex of the L protein that contains the RDRP domain, as well as VP35. It's very similar to RSV, which is also indicated here. So these are non-segmented negative sense RNA viruses, and RSV expresses the polymerase as an L protein together with the P protein. So in Ebola, the counterpart of the P protein is VP35. If you look here at single nucleotide incorporations, and you can see that the efficiency of incorporation is actually quite high, very similar to ATP, which is a good thing. You would like to see that. The higher the rate of incorporation, the higher the chances are that downstream there's some inhibitory effect. So with RSV, pretty much the same thing. And if you test for a human enzyme, it's in this case human mitochondrial RNA polymerase, which can predict toxicities also. You can see that ATP is heavily favored over Remdesivir. So that's a good thing. That just based on the biochemistry would indicate, okay, there's a chance that there are antiviral effects, and that was previously shown. And toxicities, just judging by this one enzyme, human mitochondrial RNA polymerase, could be low. And if we are looking for multiple incorporations, so the idea was to demonstrate actually that there is competition with ATP, but also we wanted to see whether there's any inhibition, because incorporation alone does not necessarily translate into inhibition. So the experiment that we used here is, you can see the template, the primer. We incorporate in red here hot nucleotide, so it's old school biochemistry, detecting the products with radioactivity, running gels, and see what you get. Here we have the competing natural nucleotides at low concentrations. And you see already here that there's clearly an incorporation of Remdesivir at the position where we would expect it. And you can see that there's a shift then in the location of the product. That is expected because the structure of Remdesivir is different from ATP. So high resolution gels do reveal these differences. Now, as you increase the concentration of the competing natural nucleotide, you see that incorporation of Remdesivir is diminished. So that indicates clearly that there is competition, but it's still pretty good. If you look here at concentration of around 30 micromolar and here natural nucleotide 10 micromolar, there's still a lot of incorporation of Remdesivir. So that's a good sign. In terms of inhibition, So once it goes in, that is shown here, you see that there's no direct chain termination, which is demonstrated here with the control nucleotide. You actually see that RNA synthesis fades off and is at this position I+5. So 5 nucleotides after the incorporation of remdesivir, it gets terminated. So this is what we refer to as delayed chain termination. And the mechanism obviously must be different from the classic chain termination that I described for AZT. So this is what we've done with Ebola, and then there was a clinical trial also in the aftermath or during the 2018 Ebola outbreak in Africa. And the conclusion was that 2 antibody therapies did better than remdesivir in reducing mortality. But there was an effect also if you compare remdesivir with the non-treated population, but they were not included as an arm in this clinical trial for ethical reasons, obviously. So that was a bit of a bummer, but I will comment later on this. Obviously, this clinical trial provided a lot of other very important data for the pandemic, and safety data first and foremost, because it was tested in humans in the context of a randomized clinical trial. But if I go back a couple of years, and there were a bunch of papers looking at the spectrum of antiviral activities of remdesivir, and it's not only the filoviruses like Ebola and Marburg that show in cell culture low EC50 values, but also the coronaviruses. And that was really demonstrated in several papers by Tim Sheen, Ralph Barrick, and Mark Dennison. There's also activity against Nipah, measles, Hendra. So these are related to RSV and Ebola, roughly because these are non-segmented negative sense viruses, and the RDRP shows structural similarities as far as is known. Flaviviruses, there's inhibition as well to certain degrees, but there are certain viruses, and especially these segmented negative sense viruses, Lassa, Crimean Congo, there's no significant antiviral activity, or the EC50s are pretty high. Here also positive chikungunya alpha viruses, the EC50s are also very high. And then came the pandemic, and the big question was, what is available on antiviral drugs? And the first case in the US that was reported here, there was a case report in the New England Journal. This patient got treated with remdesivir, survived. Obviously, it's a case study. It doesn't mean a whole lot. But this was administered or provided, the drug was provided through the compassionate use protocol. Then the first data with the new virus trickled in early 2020. This is a paper that was highly cited, and they just took a bunch of inhibitors, including chloroquine and also favipiravir, that was considered at that point in time as a broad spectrum antiviral that could work also against SARS-CoV-2. And the bottom line is here, I just show the in vitro efficacy. It's submicromolar for remdesivir in vivo cells. Okay, so that was the first published, to my best knowledge, the first published data on the antiviral effects of remdesivir against SARS-CoV-2 in cell culture. we, at around the same time, we expressed the enzyme. And I guess everyone is aware this is a complex of NSP 7, 8, and 12. And I would really emphasize that this was not trivial, and the field really benefited from observations made by Bruno Canar's group years ago on SARS-CoV-1. So he figured out that you need actually the three components. It's not only NSP12 where you have the RDRP activity located, you really need NSP7 and 8 as well. So we basically used the same approach with a different expression methodology. We used the baculovirus system here and expressed everything together. And we were able to pull out the complex. So that was early 2020. And then we looked at the mechanism of action of remdesivir. And this is what we figured out. So the polymerase here shown in blue and with the primer template. And the first step is that remdesivir needs to compete with ATP. And the competition is actually pretty good. As I showed already for Ebola, it's even more pronounced here with SARS-CoV-2. So remdesivir is actually better incorporated than ATP. That was one of the most important findings biochemically. In terms of inhibition, we also see delayed chain termination, but this is very specific. So remdesivir goes in, and then we see 3 more nucleotides are added, and then we have a stop. The third observation, however, is that this is not irreversible, and inhibition can be overcome as we increase the concentration of nucleotides. So this is a hint that there is a mechanism with remdesivir that bypasses or evades the exonuclease activity that is present in these viruses. So that is NSP 14. Other nucleotides were reported in the context of other viruses, other coronaviruses, that the exonuclease activity really diminishes the potency. And that was nicely shown by Mark Dennison's group. Now, what is the reason for this delayed chain termination? And we did with the Gilead people modeling studies, and everything pointed to the serine at position 861. So we use site-directed mutagenesis. Of course, we started with an A with alanine. We couldn't see much of a big difference in terms of the delayed chain termination signal here. So we went even a little bit smaller with glycine. And then finally, we could see that the inhibition is not present anymore, and there's full-length RNA synthesis. Here, just some pausing sites that are amplified because the enzyme is a little bit weaker than its natural counterpart. That mechanism was confirmed with structures here, for instance, from Patrick Kramer's group, and they had several structures with remdesivir incorporated, and then looked for the steric clash, and that would happen at exactly at the position that we predicted, and that is modeled here, the steric clash. So there's good evidence that delayed chain termination is mediated through this residue, through this serine 861, but it can be overcome, as we can also see here on this gel. So the higher the concentration of the next nucleotides, the less we see inhibition here. So, and that led us then a little bit later to the question, so what happens then? If inhibition can be overcome, that would indicate that you actually synthesize copies of the genome that would contain remdesivir incorporated. So what happens then if we synthesize the next strand? And what we have shown is that there is inhibition again as the remdesivir, so the A analog remdesivir is copied, and this inhibition is even more pronounced. So we think the dominant mechanism of action is mediated through remdesivir in the template and the incorporation of the complementary U that is indicated here. Eventually, this can also be overcome, but you need much higher concentration of the next nucleotide. Modeling, this is not a structure, just modeling suggests also that there is a steric clash with the backbone here of so-called motif F that is conserved with polymerases and the 1' cyano group. So what are now the biochemical parameters that determine antiviral activity of RDV? I'm cognizant of the time. If I can have like 5 or 10 more minutes, if that is okay, I think I should be good then. Thank you. So I mentioned already that the competition is really in favor of remdesivir triphosphate, and it outcompetes its natural counterpart. This can be measured as selectivity. So that's basically the efficiency of incorporation of ATP over the nucleotide analog. And if the value is below 1, that indicates that the analog is better incorporated. That was the case with enzymes SARS-CoV-1, 2, and MERS as well, pretty much the same value. So all of these enzymes incorporate remdesivir very well. Now, with other nucleotides that were considered at around that time, that was early 2020, ribavirin, favipiravir, and also the HCV selective inhibitor, sofosbuvir. So anything that was available in terms of nucleotide analogs were considered. But you can see already here, if you just look at the values here, the selectivity values, they're way above 1,500 here for ribavirin and favipiravir and other nucleotides as well. That indicates that these nucleotides are not efficiently incorporated, and they may not work because of the diminished rates of incorporation. Now, along the same lines, we later looked also at the other viruses, and we could see and demonstrate that for viruses where remdesivir shows really decent antiviral effect, including here the non-segmented negative sense RNA viruses like Ebola, RSV, we always have really good selectivity values, selective incorporation. This is highly pronounced with coronaviruses, but you can still see this with Ebola and RSV. And for those viruses where remdesivir does not work, we just hardly get it in. It is not very well incorporated, and that is demonstrated here for Lassa, Crimean Congo, and flu. Now, that was in May 2020. Dr. Fauci and Birx, they announced the ACT-1 clinical trial with remdesivir that indicated a significant reduction in the time of recovery from 15 to 10 days in the remdesivir arm over placebo. But then you had also apparently conflicting clinical data, and that was with the WHO trial. And the design was different. This is generally accepted, and the goal was also different. The WHO trial, they wanted to see whether remdesivir can be used in various settings, and that obviously made a comparison also between the two trials really difficult. Later, of course, it has been shown, and this is widely accepted now, the earlier you give antivirals, the better it is. And remdesivir is approved for hospitalized patients because it's an IV drug. And once these patients get admitted to the hospital, the virus load goes down anyways, and the inflammatory phase starts. And then you just need to treat it with dexamethasone or other anti-inflammatories, and antivirals may not do anything at a later stage here in hospitalized patients. Now, that concept then was addressed by a trial in outpatients. So remdesivir was given in outpatients, and it lowered the risk of hospitalization by roughly 90%. So that comes really very close to Paxlovid, that is now also used as an oral, that is used now as an oral antiviral. All of that suggests that an oral version of remdesivir would be very helpful, and this is addressed here by Gilead. GS5245 is currently tested in clinical phase 3 trials, and VV116 is yet another prodrug. It is deuterated here at the base moiety, and the prodrug approach is a bit different. This was published as a clinical trial in comparison with Paxlovid, and it does perhaps even a little bit better than Paxlovid. Very briefly, the other RDRP inhibitor that is orally available, molnupiravir. I'm not going through the details now, but it's a mutagenic nucleotide. That's the different mechanism of action. And this is also shown nicely in the clinic. The biochemical data match exactly with the data in the clinic. So you do generate GA and CU transitions. You increase the transitions in the presence of the drug and drive the virus into aerocatastrophe. This is again from the Kramer lab, also the structures with incorporated molnupiravir that also supports this mechanism. And the point of caution is here raised by Ron Swanstrom and Ray Shinazi, and they have provided evidence to show that the molnupiravir and diphosphate may be a substrate for the ribonucleotide reductase, and the DNA counterpart may get incorporated in cellular DNA. So at least they have demonstrated that there is increased mutagenesis if you apply the drug in cell culture. That is a point of caution here, and I go back just with the last couple of slides on remdesivir. For antivirals, regardless of the nature, we need to discuss the potential problem of drug resistance. And there are some cases where resistance has been documented, mostly in transplant recipients. Mark Dennison selected for resistance in different coronaviruses, there are a bunch of mutations that show up in cell culture, and you see some of them are close to the active site, and others are really all over the place. And we looked biochemically at a very interesting one that was close to the active site, close to the cyano group, and this is the serine 759. And if that is mutated to an A, there's first of all resistance in cell culture, so increases in EC50, and we have demonstrated that you reduce the rate of incorporation with this mutant enzyme, so confirming the resistant phenotype. And the last slide here that I wanted to share, this is work from Brandon Malone and Seth Darst and Liz Campbell's lab. And this is a cryo-EM structure of the bound remdesivir triphosphate at the active site. And what I wanted to point out is the residue here that I just mentioned that confers resistance if mutated to an A. and their important interactions with the 1' cyano group. So that all of that comes nicely together. And I leave it like that here. I just wanted to mention there's one more slide with our AViDD involvement that John mentioned right in the beginning. And we are basically contributing with a library of viral RNA polymerases that we started to establish years ago. And we are always interested in adding new enzymes there. And you can see here, this is a newly discovered virus, Yeso virus, related to Crimean Congo hemorrhagic fever virus. We also have a new Nipah virus, the Lengia virus here. And that allows us to study mechanism of action, to look into new inhibitors and so forth. Most importantly, I would really like to thank everyone in my lab, and Kelvin Gordon is a grad student, and he did most of the work together with a research associate, Igor Chesnokov, and they published a bunch of papers in this area, and they worked extremely hard in the COVID, in the early years of the pandemic. And of course, our collaborators, Gilead Sciences, Daniel Porter, Jason Perry, John Bilello, and Joy Feng, Mark Dennison's group for the resistance studies, and also Ralph Barrick's group, and Tim Sheehan's group as well, and Rockefeller, Brendan, Liz, and Seth, and these are our funding sources. Thank you very much, and sorry I'm a little bit longer.